In order for the nervous system to be able to control and integrate the functions of the 10 other body systems, it not only needs different kinds of synapses, like ones that use acetylcholine or norepinephrine or GABA or serotonin, and have excitatory or inhibitory receptors. It also needs to be able to fine-tune each synapse to make it more or less sensitive, more or less active, depending on the circumstances. And this is called neuromodulation. Neuromodulation takes place in several ways. There is presynaptic modulation and postsynaptic modulation. Let's start first with presynaptic modulation. Now remember that in any synapse, the first neuron, the sending neuron, is called the presynaptic neuron, and this is the postsynaptic neuron. And presynaptic modulation, there are a number of things that can happen. One is called presynaptic inhibition, and it's where another neuron releases neurotransmitter that makes the membrane near the axon terminus more negative. And if this membrane is more negative, then the voltage-gated calcium channels that cause the release of the vesicles containing neurotransmitter can't open as easily, and therefore less neurotransmitter gets into the synapse, less neurotransmitter gets to the receptors, and the local potential in the postsynaptic cell will be weaker. All right, so that's one way. And that's what's written here. Hyperpolarization makes exocytosis more difficult, so less neurotransmitter is released. Now, there are also synapses that I haven't drawn here that release neurotransmitters known as neuropeptides. These tend to influence the synapse in the long term, over days, weeks, or months, and they influence things like the amount of neurotransmitter made. So if this neuron is not able to synthesize as much, or it synthesizes more neurotransmitter, that's going to influence how much neurotransmitter is released into the synapse, and therefore how strong the signal is in the postsynaptic neuron. So those are examples of presynaptic modulation. Postsynaptic modulation can influence things like the number and sensitivity of the receptors. Again, this is done by synapses with neurons that release neuropeptides, longer acting influences over the synapse. But a neuropeptide can either put more receptors in the membrane and therefore make this cell more sensitive and more likely to have an action potential, or it can put fewer receptors in the membrane and make this neuron therefore less receptive, less sensitive, and less likely to have an action potential. The other thing in postsynaptic modulation that can be influenced is how long the neurotransmitters last inside the synapse. So neurotransmitters, once they're released, they have to be gotten rid of. Excuse me. And there are a number of ways that this can happen. One is that the neurotransmitter can simply diffuse away. This is less common. Another is that the neurotransmitter can get reabsorbed by the presynaptic neuron so that it can be recycled and reused. And another is that the neurotransmitter can actually just be destroyed or degraded in the synapse, and then it can be reabsorbed and put back together to be reused. So neuropeptides can influence how fast the neurotransmitter disappears or how long it stays, and therefore influence the strength of the synaptic transmission. You may be familiar with a type of drug called selective serotonin reuptake inhibitors, or SSRIs. They're antidepressants that are very commonly used. These take the neurotransmitter serotonin, which would normally, after it's released, be brought back into the presynaptic neuron to be used again. But a serotonin reuptake inhibitor leaves the serotonin in the synapse where it can continue to stimulate the postsynaptic neuron, and that helps to elevate a person's mood. Okay, now I want to use one specific example of a neurotransmitter and demonstrate how neuromodulation would work. So let's pretend that this first neuron, the presynaptic neuron, is one that uses the neurotransmitter acetylcholine. So a presynaptic inhibition of this neuron would make it less likely that acetylcholine would be released. That's one thing that could happen. Or the acetylcholine, once it's released, can bind to a larger number of receptors or a smaller number of receptors. Or in the case of acetylcholine, there are enzymes in the postsynaptic membrane called acetylcholine esterase. The E stands for esterase. The whole thing just spells ache, but it's not what we call it. But acetylcholine esterase is the enzyme that breaks down acetylcholine. So one of the things that can be influenced is to have more or less acetylcholine esterase in the membrane. If there is less acetylcholine esterase, then there will be more acetylcholine remaining in the synapse. If there's more acetylcholine esterase, then it will break down the acetylcholine and stop the transmission more quickly.